So we're here to talk about synthetic biology, to talk about ideas, to talk about the world, to talk about possibilities and opportunity, I think. And I'm again really wishing to engage people as you wish in terms of approaching that. Whether you would prefer to get some background, or you have ideas you want to talk about, or anything like that. So before I get really spun up, I'd like to invite everyone who's willing to share what your wishes are for our time together, and what you most want to get out of the next bit of conversation, please.
Yeah, actually, I think we're a small enough group to where it might be helpful just to do quick intros with everybody. Drew, does that sound good?
Whatever people want. Yeah, let's go.
So yeah, if we could just kind of go around and do some quick intros. I know we did a little bit of this, so I recognize some of y'all faces from yesterday, but it'd be great to kind of make sure that we can know each other's names and pronouns, things like that.
Yeah, but yes, and how about
Yeah.
how about we go around and say, like, what do you want? Like, if you could get one thing from our conversation now, just like blurt it out. Like, I want this, I want that. Okay.
Yep.
My name's
Sounds
Drew.
good.
I use he, him, and I want to know what you want.
Good. Drew, do you want to popcorn it to someone?
Popcorn to Carlos.
Hi everyone, my name is Carlos, he, him pronouns. I'm pretty new to synthetic biology, so I'm honestly okay with anything. Yeah, anything new I can learn. Yeah, I welcome it. Yeah.
Carlos, popcorn.
Popcorn to Sophia.
Hi, I'm Sophia. I use she, her pronouns. Oh, sorry. I'm a freshman, and I'm just like Carlos, very new to synthetic biology. And I kind of just want to be more exposed to the field. And I'm like I like solving problems. So I want to like be exposed to some problems that are seen in synthetic biology so I can get more ideas of what I can like explore in my project. Yeah. And I'll popcorn to Montana.
Hi, my name is Montana. I use she, her pronouns. I'm currently a sophomore. I'm also new to synthetic biology, but what I think is really interesting is particularly like what is the future of synthetic bio? So like 50 years down the line, like what do you think we could be doing with synthetic bio? And then also the intersection with bioengineering and ethics. So what do you think from other standpoints, whether that's government or academia, how do you think the ethics of bioengineering are going to catch up with the sort of vanguard technologies? I will popcorn to Alan.
Hey everyone, my name is Alan. I use he, him pronouns. And I want to know what's the most exciting thing that you've ever done in this field. I think that'd be pretty interesting. I'll popcorn it to Audrey. If Audrey's unable, it seems like her mic is off, so I'll popcorn it to Allison.
Hi everyone, I'm Allison. I use she, her pronouns. I am here because I want to know why you chose this path, and then also like what I guess advice do you have for undergrads? And I'm also here because I just want to know more faculty at Stanford. Oh, and I'm a freshman. And I'll popcorn it over to Finso.
Sure, thanks, Allison. Hi, everyone. I'm Fin Sam, and he, him, his. And what I want to know is, what are some interesting things happening with SynBio in space, outer space? I think that's an interesting area to go into, and what can it do for like space exploration? But I also want to know about SynBio in general, as I'm also fairly new to the field. But yeah, I'll popcorn it over to Aaron if you haven't gone yet.
Yeah, hey, I'm Aaron. I'm a senior, he, him. I'm actually a physics major, but I've taken one systems biology class. I found it really compelling. And I kind of want to hear about like some of the different paths people have taken into bioengineering, synthetic biology, systems biology. Oh, and I'll popcorn to, who hasn't gone?
I haven't gone.
Oh, yeah, China.
Yeah, hi, I'm China, she, her pronouns. I'm a bioengineering major, a graduating senior. And what I'm hoping to get from this is to learn more about where SynBio is going. So yeah. And I could popcorn. I think everyone's went.
Audrey, Audrey was having mic issues, or?
Yeah, Audrey, are you there?
Yeah, so on the participants list, it seems like the microphone symbol just isn't there. So maybe,
Okay.
yeah.
And Lauren and Sarah, you're good as our hosts or co-hosts, is that right? Or do you want to chime in?
It's us and Ayush and Alan as well.
Okay.
But I'm excited to hear more about essentially ways that we can ideate solutions to problems, like global problems, and how we can use bioengineering to fix them, and just kind of the ways that we can come up with those ideas.
Mm-hmm. Sarah?
Yeah, I'm interested in hearing like what kinds of things are you looking forward to doing yourself in your future career? Like what kind of path are you looking at going on in the future?
Okay. Great. Is anybody else? Carlos, Sophia, Montana, Alan, Allison, Finsam, Aaron, China, Lauren, and Sarah. Thank you so much for sharing. Let me try and pick up and then we'll just play and run, huh? So, Allison, you were asking about sort of why? Oh, hey, let's see. Well, Allison was asking about why for me, and everybody's different, so I'll just share a little bit about me and my weaknesses. I have a hard time with discipline, like the mind controlling the mind and like staying focused on something. And so for me to be successful, I've had to learn how to overcome that weakness and what makes me sustain effort to do something to get to success. And in understanding that about myself, which I don't think I'm still understanding myself, what I've learned is I will do something if I'm having fun. So personally, like if I'm having fun doing something, I'll keep doing it. And then the other thing I've learned is I don't have as much money as I need to do the things that are fun. I'm not independently wealthy, right? I grew up in Pennsylvania. My dad didn't go to college. He was a successful shopkeeper. He ran a luggage shop in the suburbs of Philadelphia. And so like for me to have fun and do things, I need to also be doing things that somebody else is going to pay for. And the simplest way to abstract that is I have to be doing things that somebody else thinks are important, right? And so I've been running this algorithm of what's at the intersection of fun and important that I can find, and anything that's at that intersection, I will do it and it will be important and it'll get resourced, right? So I've just been running this abstract algorithm. I wake up in the morning, I'm like, am I having fun? And am I doing something important? Now, for me, it's been fun always to tinker and to build. Right? It's like the cliche, like I like to build things. When I grew up and went to college, I studied, well, where do you study building in engineering? It's like civil engineering, like literally building buildings and roads and water supply and sewers and stuff like that. So my undergraduate degree was in civil engineering. I spent a summer doing an internship with Amtrak on construction management for the Northeast Corridor train lines between Washington and New York. Go into 30th Street Station in Philadelphia and take the train each day one summer to a different site for construction. What I just fast forwarding into bioengineering and synthetic biology, it becomes apparent that biology is a way of building. It's nature's way of building in particular. Of course, you could say tectonic plates are nature's way of building mountains, but for the living things, and everything that the living things do, like make oxygen and so on, it's the living things that are building it. And so when I'm running, what I'm trying to share is this algorithm of like what's fun and important, what's fun and important, what's fun and important, like waking up every day over and over again. The cool thing about that algorithm is it lets me change who I am. So I was a civil engineer, and then I was an environmental engineer, and then I became a bioengineer eventually, as that became a thing through synthetic biology. So like running this general purpose algorithm hasn't locked me in. It just lets me keep growing and going. And then that allows for this discovery of biology as a way of making, nature's way of making, and so on. So Allison, I hope that's a little bit responsive to your prompt. Do you want to chime in on that?
Yeah, no, that was really great to hear, and pretty much answers my question. But yeah, yeah, I'd love to hear more about like your experiences and
Yeah,
like challenges and stuff.
yeah, and well, one thing I would say is here's something on background. When you're doing something new, and synthetic biology has plenty of new, and we'll get to where I think it's going and what I'm excited about, but more broadly. When you're doing something new, what that means is it's actually new. Like new is actually new, and that's exciting. But I want to give you a particular heads up. When you're doing something new, the newness of it means that other people won't necessarily know about it. Other people won't necessarily see it for themselves. You will be inevitably, if you would go into this direction, and I know some of you already are, right? As you go into this direction, you will encounter the new, and the newness of it will require you to stand up for the new and to teach others the new. Let me give you an example. Instead of just using these words over and again, let me just give you a practical example of how this will show up. Where are you going to find the job postings for the jobs doing the new thing? For a job posting to come into existence, what that means is somebody else, somewhere else, has to figure out that there should be the job, and then they have to get their colleagues at the existing institution to agree to create a new job, create the position, run the search advertisement, collect applications, interview people. Most jobs that are posted are not like that, right? Meaning not compatible with the new is what I mean to say. Rarely you can find places that are hiring, like we want to hire somebody to do the new innovative thing, right? But a lot of corporate America and corporate world, it's like we need somebody doing the new thing. No. We need somebody doing the thing we already know we need to do, right? So just like in my life, as I got into the new stuff, the likelihood that I would find my path by applying to an existing job went to zero. Right, so I just stopped applying. I stopped applying to jobs. I had to instead start creating the job for myself, right? Finding the people. Okay, now how do you do that? Just a second little practical advice. A lot of times I fall into the trap of, oh, here's the big brand name person or institution where I have to get a job or I have to work with them. And that can be very hard because that big brand name person or institution is already going to be really busy. The other thing to do instead is to find the people who want to work with you. Because there's going to be people who want to do the new thing. You may not know who they are, and they may not know who you are at the start. But when you find the people who want to work with you, then it's very easy to work together, as opposed to trying to convince somebody who's already installed and doing something important to change what they're doing to work with you. That's almost never going to work. Again, not in my limited experience. Okay, let's get back to synthetic biology, I guess. What are we talking about? We're talking about an increasing capacity to express human intentions within living systems, an increased capacity to partner with the living world to do things. This can be a one-way partnership where we, the engineer, are telling the biology what to do, which is a lot of what's happening, like make the medicine, right? Or I think equally interesting is to think about it as a symmetric partnership within the context of resilience, sustainability, ecosystems, and flourishing, not just humans, but all life on Earth. That symmetric relationship, I think, is weaker culturally and politically relative to the commercial asymmetric. We're telling biology what to do. We're, quote, programming biology. I just want to note that. Lauren, I'll take your advice. Do you think I should show a slide or not? I'm just looking at my scratch pad and the comments about on-ramping and newness. Some of you
I'm a fan of slides.
Okay. Okay. We'll try it. We'll try a few. All right. Do these come across?
Yep, we can see them.
Let me hit you with this. I hope everybody knows the name of Martin Luther King Jr.'s famous speech, right, about emerging technology, right, about AI, Internet of Things, 5G wireless, big data, blockchain, robots, drones, 3D printing, gene editing, solar photovoltaic, right? Like, no, that was not about this, right? His genius was, his speech was not about a dream. So I use this as a way of hopefully motivating you for yourselves and others to think about this question. So what are our positive dreams of and with biology? If we exist at a time where Stanford's created a bioengineering department, it's coming up on, still a teenager, but it's growing up as a department. What that means is we're getting better at engineering biology in this century. We're going to get really good at it. And so it begs this question, like, so what? To what end, right? And that's not for me to decide, but certainly for me to ask you, to encourage you to decide. And I'll give you an example of how I fill in this image. By the way, what is this image? You might be wondering. Like, remember, I'm a civil engineer long ago. This is a double-decker suspension bridge grown from the roots of a rubber tree, apparently, right? It fixes carbon as it grows. Very different than concrete. So here are some facts if you haven't thought about biology as an engineer might. Biology is all over the place on this planet. It's powered by light, photosynthesis. How much energy is biology harnessing? The answer is 90 terawatts. Why does that number matter? Well, civilization's running on 20 terawatts. So the energy powering biology is 4.5 times what civilization consumes. Right here, the napkin math suggests that we should be able to make everything 10 billion people need without trashing the rest of the planet. If we're not doing that, there's something wrong with how we're configured to make the stuff we need. And by the way, this number 90, as you'll see in a moment, in response to some of the things I'm most excited about, this number 90 is going to go up. There's no ceiling on this 90, it turns out. What else can we say about biology? It reproduces, it grows, it heals, it's very functional in terms of interacting with other elements and materials. And the cultural aspects, the political aspects, the ramifications are off the charts. So now comes we could discuss the things on the left if you want, but I don't really think these are debatable. These are just sort of, I'm going to claim these are facts, right? The thing we should debate and discuss and argue about for as long as we can is what goes on the right. So what do you put in on the right? Here's my placeholder, but I'm very happy to upgrade or change. I'm just giving you as an example. As mentioned, we should be able to provide for ourselves. We should be able to stabilize and recover biodiversity instead of driving things to extinction. We should make infectious disease obsolete. COVID-19 is a fine example for why we should do it, but we should do it. I'll note, right, that 2 of the best vaccines for COVID are synthetic biology-based vaccines, synthetic nucleic acid vaccines. Is the bioeconomy going to happen to us, or are we going to happen to the bioeconomy? Are emerging technologies things that happen to us, or are we things that happen to emerging technologies? Right. So that's what I mean by a culture of citizenship. I have a bias there. I want to be a citizen of the bioeconomy. I want my lineage, my kids, to be citizens of the bioeconomy, not consumers or subjects or objects. So how we relate to this technology. Okay, so I'll drop out of that slide. Let me give you some cool examples, actually, and then I'm actually just going to push. Do we go to 11:30? Okay, so I'm just going to push for another 6 minutes. Here's a cool synthetic biology vignette from Northwestern, a friend, Mike Jewett, and his team. Like, what if people could just make vaccines where they are? And so this is freeze-dried cytoplasmic extract. You just add water, and wherever you add the water in the vial, the vaccine's made. This is for a type of vaccines called conjugate vaccines. So synthetic biology success from the last few months. And they're showing that it works in mice in this academic paper. So that needs to be translated, right, into humans. Here's a Stanford project that's come out of Stanford, right? So this is a cartoon schematic of yeast, the baker's yeast, brewer's yeast. The chemistry here is not natural metabolism. This is bioengineered metabolism. And so this yeast takes in glucose, sugar, and in this case, spits out scopolamine, which is used for treating Parkinson's disease and motion sickness. So normally this medicine would be sourced from a nightshade plant grown in plantations in Australia. But now you could make this medicine in a fermenter anywhere you can run the fermenter. And what I love about this image is it's showing where synthetic biology has gotten to in terms of the scale and sophistication of metabolic engineering. Like these engineers, Prashant and Christina, are now able to engineer 30 plus enzyme pathways inside yeast by recapitulating the spatial morphology of a complex plant into the microscopic organelles within inside a single cell, routing the chemicals around the different locations to do the different chemistry. If you want to think about this differently, just strategically, what their work implies is that any natural product, any chemical made in a plant or microbe found in nature could instead be made by brewing and fermentation. All you have to do is you have to re-implement the biosynthetic pathways inside yeast or an equivalent organism. So that's going to change the world, right? It's literally going to create new supply chain possibilities and so on. That's in the immediate. The longer term is, if you look at how supply chains supply people, typically they only supply 2.5 billion people, like the big global supply chains. So the majority of people who need medicines don't get medicines. And so the longer term promise of synthetic biology would be to take this type of technology and not have it be in an industrial fermenter in a big tank in Minnesota, but a distributed biological manufacturing process that anybody anywhere could brew up what they need, right? So making what you need where you are is, I think, the ultimate promise of biology as a complement to centralized provisioning driven by an industrial economy. You can take wood chips and make leather surrogates. Just like build with biology. What does this actually look like? It's like build with biology. There's a cool example on the MicroWorks website of a new handbag they've made in partnership with a French fashion house. Lauren et al. And Sarah, I hope you appreciate what's coming. Like, what's salmonella? Here's a question. For people who are new to synthetic biology, what's behind this is an exercise called unnaming. Like, take the names away from things and use that to reimagine what things could be, right, as a tool, right? So synthetic biology, I want to say something in a strange way. One of the things I love about synthetic biology is it is antithetical to the status quo. And it promises opportunity via quid pro quo, taking something and replacing it with something else. But to unleash this, you have to understand what the status quo is and how to navigate into the possibility. So one tool is this tool of unnaming. Like, you might think of salmonella as this dangerous pathogen that'll give you diarrhea and could kill you. That's true. But if you just pause for a moment and say, well, what is salmonella? Well, it's a microbe that gets inside me. Maybe it could do something. What if we called it salmonella? What if we figured out how to bioengineer salmonella to not cause diarrhea, but to deliver a DNA vaccine? Right? And so there's a neat paper from Roy Curtis 9 years ago showing how to vaccinate mice against flu using salmonella. It's not marketed very well, like they're not calling it salmonella. I'm not sure that's the best name. So the cultural product design aspects of this definitely need lots of opportunity. Here's another example. What is a sinus infection? Oh, it's painful, right? Well, what if you had friendly microbes living inside you telling you what was infecting you? What if you have a seed? What's it called? A self-replicating embedded environmental diagnostic? Is that right? Sarah, Lauren?
Yeah, self-replicating, environmentally embedded diagnostic.
Yeah, so like I like this idea a lot. You know, like why do I have to go somewhere else to get diagnosed if I have an infection? Shouldn't the color of my mucus just tell me what's infecting me? Like a coronavirus causes bright orange mucus, an influenza virus causes bright purple mucus. I really would prefer that, right? I want real-time, runtime diagnosis. Okay, I'm just gonna give 2 more slides, maybe 3. Oh, I'm just gonna do 2. So here's something that's super exciting to me. There are 2 postcards here, and I'll walk you through it. On the left, the chemical engineers and material scientists have figured out how to split water with electricity, and then in a simple catalyst-based process, fix carbon dioxide from the air to make a single-chain organic molecule called formate. Single carbon organic molecule called formate. So just note, you can go from electricity to a single carbon organic molecule, electricity to formate. More recently, the metabolic engineers enabled by synthetic biology have re-engineered metabolism of microbes to grow on formate as opposed to glucose. All right, so if you put these 2 postcards together, the left one says you can go from electricity to formate, and the right one says you can go from formate to biomass. Now you basically go from electricity to biomass, if you can put these 2 things together. And at the bottom, I have some napkin math that Tom Jaramillo at Stanford Chemical Engineering helped me work up. 1 kilowatt hour of electricity, or about 10 cents of electricity, makes a gram of biomass if you did this today. But his research team thinks they can increase the efficiency of the left side to make this go up to 30 grams of biomass per kilowatt hour. Now that's really interesting. Like even 1 gram is pretty interesting because a gram is enough to make a dose of a medication. Instead of feeding a fermenter with glucose that I have to get from somewhere, I could feed a fermenter with electricity and air, which is bringing in the carbon dioxide. And that's really interesting because of how I can source electricity through photovoltaic, solar, or other, right? And I won't belabor it, but the way electricity generation is going, we're transitioning to electricity generation abundant civilization. So there's going to be more electricity than we know what to do with. The utilities will keep increasing their prices because that's all they know what to do. But from the physics perspective, there's going to be plenty of electricity. What does this lead to? Well, who knows? I think it could lead to the following object that I'm showing in abstract cartoon form only. You've heard of the personal computer. How about the personal biomaker or the personal biosynthesizer, the PB? And this box takes in electricity, internet, which is the biocode, air, which brings in nitrogen, carbon, water. We're going to have to figure out, we're going to need print cartridges for other elements needed. And what comes out of the box is anything that biology can make. Like whatever biology could be programmed to do. That's what I'm excited about, in part from like a technical perspective. I want to return to Aaron. Are you still here, Aaron? Yeah.
Yeah, I'm still here.
Yeah. So I want to return to your comment, which I questioned or prompt at the start, which in part I took as the combination of immigration into a new domain of knowledge and coming from a physics background, also within the context of the now. So do any of you have like a subscription to HBO Max or Netflix or Disney Plus? Everybody knows like online streaming content. Okay, when you download a show, and you're going to stream a show rather, what happens? Like if you go onto the internet and one of these streaming services, what happens? You're like, oh, I'm going to watch Mandalorian or whatever your favorite show is.
Well, like you send a request from your client computer, basically, it goes to a server, and the server then continuously downloads the show to your device, right?
And does it work when you do that?
Most of the time.
Yeah, exactly. Almost like by most of the time, like 99% of the time.
Yeah.
Cool. Right. So here's a cricket player from the Indian national team, if I remember correctly, and he's relaxing in quarantine in Australia, and apparently he's on I don't know what platform he's on. Is that Netflix? I can't tell. So one of the things that's a challenge in synthetic biology and bioengineering is our biocode doesn't always work. Like whereas if I'm going to watch a program with my kids, I want to stream something. I don't want to download a research project, right? At least for evening entertainment, maybe for learning. I want to just download code that works. So I would say we're pretty, for most of bioengineering and what biology could build, we're pretty far away from that possibility. There are counterexamples. So the best counterexample in the contemporary would be the COVID experience, where in or around January of 2020, researchers in China uploaded the DNA sequence to the COVID-19 genome on the internet. And then elsewhere, people downloaded that sequence information and remade the COVID-19 to study it and to make the vaccines. And so that's what I would call the beginnings of the bionet, right? Where the superpower of the internet that allows for information to be decoupled from a position in space-time, and then the information can be recoupled to another spot in space-time. That's beginning to happen for biology. So if you think about the personal biosynthesizer and the idea of getting to distributed biomanufacturing so people can build the biology they need where they need it, for that to really go, we need to make the bionet happen. For that to work, we have to leverage the ability to sequence DNA, turning an abstract representation of the physical genome into information, and then move that around. And then if we change it and recompile, it's going to work. So again, I think the COVID-19 and the vaccine is an example that this isn't impossible, but it's mostly not true for most bioengineering projects. Like, it's mostly hard to build the DNA, and when you build the DNA, it mostly doesn't behave as you expect. So I would suggest that there are some fundamental puzzles that we should deal with to really move the needle on this. The best example I can give you is the fact that there is no cell on Earth that is fully understood. So the simplest cells in nature have about 450 different genes, each gene encoding a function essential for life. And the best studied simple cells, there are about 100 of the 450 genes. Nobody knows what they do. Yet each gene is absolutely essential for that organism to be viable. Don't get hung up on the genes. Think about them as functions, because the genes are not typically doing stuff. It's the functions they encode. So an essential gene of unknown function means that there is a physical function essential for life. It's a physical function that's unknown. And this problem has been around for 2 decades now. So the questions about where am I heading and what am I doing, I'm working on that problem. And let me see if I can show you what that looks like. So this is work done by Eric Way and Anton Jackson Smith and Akshay Maheshwari. These are bioengineering and electrical engineering PhD students who've been laboring quietly for 6 years to try and figure out things like, how do we build a cell from scratch? Could we do that? Imagine biotechnology if the practice of building with biology is an Edisonian tinker and test where you have to try out 1,000 different prototypes to get it working. But we understand cells so well, we can build them from scratch and we know how to operate them. And then it's much more of a straightforward rational design endeavor. Yeah. So let me show you what this looks like as of, what month are we in? This is 2 months old. Okay. This hasn't been published yet. Do you see this little gold-like shell thing, strange thing? All right. So I'm just going to let this run. So this is a computer false colored image of a sample that was observed on the electron microscopes up at SLAC, the national lab. This work was done by Anton Jackson Smith, a BioE student, working with Hua Chu and his research team at the national lab. And this is an object, the gold shell that's cut away is about, the whole thing is about 340 nanometers in diameter. So just a little under half a micron, and we built it. We built this thing. And the gold shell is the lipid bilayer, the membrane, and the purple things are ad hoc organelles inside the thing. And the red blobs we think are the 50s subunits of the ribosome. The ribosome's the thing that makes proteins. And then we're stripping away from the image all the other molecules inside this thing. But collectively, we know every molecule making up this object, and this object, if you put DNA into this object will read out whatever DNA you put into it, making mRNA and protein. All right, so this is a precursor to a fully synthetic, built from scratch cell that we're building bottom up. Does that make sense? The puzzle we're now confronting is, what DNA do we add to this object to get it to grow and divide? And if we can answer that question, then we will have not created, we will have constructed life from scratch. And if we can do that once, then we can do it again and again and again, and help transition all of bioengineering to more rational engineering. Does that make sense? So this is like fundamental synthetic biology research. Like, the cell is the fundamental unit of life. Let's learn how to build cells. If we can learn how to build cells, it's game on. In the absence of that, what you see is things like in the absence of that, what you see is things like Genko, right? So so here's a website for a company. The company's called Genko Bioworks. They're the organism company, biology by design, most advanced manufacturing technology. We program cells to make stuff. And so this is, I'm using this example because this is an example of a company created by students, right? Austin and Reshma and Barry and Jason were students, some with me, some with Tom Knight, but they and Tom founded and lead the company. And what's interesting, though, is if you learn about their company, they're building these really big foundries because the engineering of cells is hard. And so they've got to scale up tinker and testing, right? So they're like apparently this is how they represent themselves. We've got robots, we've got people carrying around lots of samples, and one person at Genko can do the work of 10 people somewhere else, right? Or something like that. So this is an artifact of the reality that we don't understand cells completely, that engineering of biology is hard. So my job as an academic is to build the foundations that make the engineering of biology easy, right? Through the fundamental R&D. I want to show something really cool. So all right, see if this comes up. Okay, this will be the last thing, then we'll go. Okay, so I put this tweet up, this one here 2 days ago. So in 2005, I write, struggling as your best and brightest, labor for 3 years to manually build part of a designer genome. Wired 2020, 2021, one pod assembly of the same genome. And I'm linking this preprint here. So let me see if I click on this. Does the screen share come along to the new image? Do you see this preprint? So check this out. Here's a paper, hasn't been peer reviewed yet, but looks really good. Rapid genome construction from 52 parts. And this genome's not as big as a human, it's only like 40,000 letters, but this is enough to build any virus you want, almost, right? And they're basically saying, we figured out some new tools, and we applied these insights to, in a single reaction, build bacteriophage T7 and get working biology from scratch. So they're reporting how to build a 40 kilobase fragment of DNA. If you don't, that number doesn't mean much. Like a gene encoding a function might be 1,000 bases. So 40 kilobases could get you 40 different biochemical functions. It's quite a lot. So you can now you can do this in one 30-minute reaction. Whereas you go back here, well, it took us 3 years, right? So like when I started my lab, doing that work was 3 years of effort from the best and brightest PhD students in the lab, and now it's 30 minutes. Let me see if I can find something in the reply. Oh yeah, like here's Shri, he was the student, right? And like here's the method section in his manuscript. Like this is 3 years of work to build a little fragment of DNA, right? And just keep going. Hopefully it's not too offensive. Nope, that's not the one I wanted to find. There was a really neat, well, and anyway, I'll just leave it. All I'm trying to communicate is we're transitioning to a future called Get Me the DNA, as opposed to I have to manually construct DNA using some ad hoc method. That future is not equally available to everybody, right? So Sarah, for example, is building DNA, but sometimes sourcing DNA from companies. But that paper is so interesting to me, just a few months old, because it really does suggest how to build DNA to make any DNA you want. What does that mean for you in terms of what you should do, right? If you want to be a bioengineer or you want to be a synthetic biologist, I would say it becomes ever more important to figure out what to build and why versus how to get it built, because you can always engage another service to build the thing if it's important enough, right? So thinking about what biology could be and what we wish for our relationship with biology, being a designer, being a critical thinker about what's needed in the world becomes way more important than the technique of making the detailed molecular biology, if that makes sense, right? So I would really, that transition has been a long time coming, right? So it's like, I'm bookmarking something from 2005 and 2020, so it's 15 years coming, but it's not stopping, it's not slowing down. That transition is now arriving, and that I think really should be something you confront. I don't like prescribing, but thinking about what to make as opposed to how to make it, like, becomes a lot more valuable, if that makes sense. Okay, I went into professor mode. I'm sorry, a little bit I did. So SB new, oh, we didn't get to Montana. Ethics. What do you think? Is there anything ethical in terms of aspects of stuff? Or is this all just like not to worry?
I think there has to be something ethical. Like, and I don't think you can ever remove ethics from it, especially when you're dealing with life.
Mm-hmm. Yeah. So of anything I'm presenting or anything you know about, is there something that strikes you most of all?
I mean, I think constructing a cell is really cool, and I don't know. I don't know how I feel about being. I don't know if it's necessarily about something that you're doing that I don't think should be able to be done, but the fact that it's technically maybe unregulated, or that there's not maybe like a global consensus, or like can we even reach a global consensus? And who should be in charge of that? I think that is maybe more intimidating.
I agree. It's kind of wild. I'll just admit to it, right? Like the 1960s had a space race, And of course, that was in the context of a Cold War. So the ethics of that, is probably myopic and filtered through that lens. I think the 2020s are gonna. It's not called this yet, but I'm naming it. Like I'll at least do my shoot from the hip hot take unilateral naming. Like there's gonna be a life race, and just like we exist in a gravity well, and the rockets help us get up out of the gravity well, we also exist in a life well. The life on Earth is constrained by the life that comes before, and being the lineage basically, and being able to construct life from scratch removes that constraint potentially and lets us climb up out of the Earth's life well. What's really compelling to me is I don't see that as like the deep space vacuum of nothing. I see that as the possibility of biology to be accessed, right? So the reality of biology is very constrained in relation to the possibilities of biology. Now, again, like, do we want to trigger a life race? Should we name it that or not? I don't know, but like 2015, I didn't have any confidence we knew how to go about building a cell. 2021, I don't know how to do it, but I know we have the tooling in place where we can run the process and we'll get to the finish line eventually. I think if we sprinted, we'd get there in 1,000 days. If we don't do it, it'll never happen, but I think inevitably it's going to play out. So I think you're right.
The question would be, what do you see as the finish line? And then also, I mean, the hindsight's always 2020. Like, what do you see going wrong? And if it does go wrong, what do you think are going to be the repercussions, and on whom, I guess?
I started with MLK Jr. for a reason, and I have a dream for a reason, because that's got to lead. We've got to figure out how to answer the question, what do we wish of our relationship with biology? What do we dream of? Because like, I have a plan for building a synthetic cell is just inane, right? It's not even a relevant thing to say if we don't know where we're going and why. And I want to unpack that a little bit more. Dreams are tools for organizing people to do things. And there are there are many different types of dreams, but here are two types. The nightmare, everybody knows about nightmares, and then the positive dream. Oh, that was awesome, wow. Okay, so if you here's the deal. If you choose to apply dreams as a tool for organizing people, if you use the nightmare approach, what ends up happening is the nightmare has to start to come true, or it has to be perceived as starting to come true before you get coordinated action in response to the nightmare. Climate change is a fine example of this. It's a nightmare framing. It's now becoming the climate emergency, right? Since Scientific American just said, we're no longer referring to climate change, it's now climate emergency, right? So the irony here is horrifying, right? Like, people who are concerned about climate change have chosen a nightmare framing of their dream, which inevitably and ironically requires the bad thing to become true in order prior to getting sufficient collective action. So there's been a failure to articulate a positive dream regarding climate. And but the positive dream lets you organize people in advance, right? And MLK Jr. took the approach of a positive dream. The Apollo program going to the moon was a positive dream, right? And so, like, before we got to the moon, you could take 3% of the economy and devote it to going to the moon. So for me, I think the game is going to be one, Montana, in terms of the structure of the narrative, the story we tell, our dream of our relationship with the living world, and whoever gets that right is going to have a good time, right? But there's going to be competition for that dream. Let me give you an anecdote that like make this really operational. I was talking to a colleague at MIT, and we're both thinking about maybe serving in the Biden-Harris administration, like do some public service, right? And we're both engineers, and so we're personally happy if we get something done. Like, if I have a good day if I like ship something, I get something done and it ships. And if I can't get something done, I get really frustrated, right? So we're both like that. It's like, what are we going to do if we go? And so they were saying, well, my goal would be to create a bio belt. We had the interstate system developed under Eisenhower. Let's create the biostate system where every state is connected to the bionet and has biomanufacturing capacity, and there's fermenters for doing modern manufacturing everywhere. There's good jobs and money, right? Blah, blah. Like, great. Like let's build infrastructure for the bioeconomy, right? 25 billion a year for that. Okay, well, what about you? Like I said, well, if I went to serve, my deliverable would not be in infrastructure. My deliverable would be in culture. My wish, I wish for the nation to fall in love with bioengineering. I wish for the nation to fall in love with biotechnology. We don't have that relationship with biotechnology right now. We got GMO dysfunction, we've got all sorts of other problems, right? Like we've got a really dystopic, culture with respect to biology and how it, but my deliverable will be like, I'm shipping love, right? Like I want to figure out how to enable a culture to fall in love with our capacity to partner with life. So just like, that's what I'm blurting out these days.
So it's a marketing scheme.
It's not marketing. It's not. It's culture. So let me offer this in response. Stuart Brand, long ago of Stanford, has this tool he calls paste layers. All right. Have you ever heard of that? I'm just checking. I'm going to show you this as opposed to tell you this. All right. Check this out. South China Morning Post newspaper headline: Tycoon donates 500 million Hong Kong dollars. It's like 70 million US to the university there to start a new synthetic biology institute. Synthetic biology DNA technical engines of the century. Cool. So called up a friend there, right? They're emailing, they're recruiting faculty and students. We should all go there, right? Well, I don't know, like what's going on? So we call them up and it's like, well, we've decided where we're going to build our new building. It's on the highest point of land in the main campus. We're actually going to spend $1 billion US on this synthetic biology institute this decade. It's about the industrialization of biology. Industrialization of biology. Wow. Like that's a 19th century British metaphor around concentrated capital and manufacturing. And we're going to do that to biology. We're going to industrialize biology. All right. Like, what should we do? That's going to happen. That's going to happen all over the place. That's going to be inevitable because that's the current trope. Here's the paste layer, and this is my response to your valid claim regarding marketing. So you see this little schematic? So this is the paste layer tool from Stuart Brand, but applied for synthetic biology. So what Stuart says is a healthy civilization is operating across 6 characteristic layers from bottom to top: nature, culture built on nature, governance built on culture, infrastructure within governance system, commerce on top of infrastructure, and then fashion. And the reason he calls it paste layers is because the pace of change increases as you go up and decreases as you go down. So nature changes very slowly, hardly at all, but culture changes, and so on. Fashion can change quickly. A healthy civilization's got good stability across all these layers. But when things start slipping, it's problematic. The easiest example I could give would be, temperatures are increasing across the world, and in California, moisture's going down. People are developing housing tracts and fire zones. The infrastructure around electricity transmission's aging. Winds are coming up. Next thing you know, we have conflagrations and wildfires that are hurting many people, right? So it's like things are changing out of step with each other across the layers, and chaos results, or conflagration results. So when I talk about culture, I'm talking about not how do I market somebody's product that's shipping as a new medicine or handbag. I'm talking about how do we think about our relationship with ourselves, with other living organisms, and so on. So I'm thinking about that. And when I talk about building a cell from lifeless ensemble of molecules, I'm talking about reconfiguring nature, basically, right? So I'm hacking and shipping products down in the lower 2 levels. One thing I'll end with this, right? Because one thing I want to highlight is you can do synthetic biology and bioengineering in every level. There are bioengineering innovation opportunities in every level of the paste layer framework, right? And on the right side of this, what I'm highlighting is most of the world is focused on the commercial level. Like that's where like 99% of the activity is. Everything else is just open for innovation or for maintenance, for stability, right? For keeping things the same. There's a lot more there than I could possibly understand, let alone relate. But that's how we're thinking about it. So when we're advancing synthetic biology at Stanford, I'll just put a bow on it like this. Our strategy for synthetic biology at Stanford is to do the whole stack, right? Like partner with the Master of Fine Arts program, right? Not to market biotechnology, but to figure out what should we be thinking about constructing or designing or provoking. So do the whole stack, and there's opportunities all over that. It's very tricky, Montana, I will admit. Like, it's very tricky because synthetic biology probably sounds like a technology or a science thing, right? Because and it's like very tricky to create equity and symmetry across disciplines and tribes in co-creating something sublime. Here's another way. I'll leave it with this. You know, what was the name of Jimi Hendrix's band? The Experience. The Jimi Hendrix Experience, right? So that's really interesting. So I think about synthetic biology at Stanford in the context of the synthetic biology experience. So that by that, I think we might mean to invite people to come participate in the co-construction of the sublime. You know, that synthetic biology is not something happening to you, it's something that you can experience, and by that experience, shape it and make it something sublime. This will not happen by default, right? What's going to happen by default is industrialization. And what's going to happen by default is structuring of groups of people using nightmare dreams that trigger crisis to then direct reactions, right? So that's what you're going to get by default. Everything else is opportunity. I suggest we go do the opportunity stuff. Sorry. I'm not going to represent I got everything exactly right, but I'm grateful for the prompts, and thank you for your patience. And I'm happy to talk more. And Sarah and Lauren and Alan and Colin and Ayush, thank you for this and what you're doing. Yeah.